Hello and welcome to this Nature Research custom media webcast titled PCR-Free Whole Genome Sequencing with DNBSEQ Technology. My name is Sarah Hiddleston and I will be your moderator. Today's webcast is sponsored by Complete Genomics. There is an increasing demand for whole genome sequencing, but cost is still a barrier for small-scale projects. With broader clinical use in mind, efforts have been undertaken to further reduce cost and turnaround time, while improving sequencing performance. This webcast will highlight how to accomplish completely PCR-free whole genome sequencing via the DNBSEQ platform. We'll begin with a short presentation. Dr. Rade Dermanats is Chief Scientific Officer for Complete Genomics. He'll walk us through DNA nanoball-based sequencing technology and the DNBSEQ platforms. He'll also discuss how these technologies are helping to move whole genome sequencing into a new era of broad clinical use. We'll then move on to a question and answer session with you, the audience. You can ask a question at any point you wish throughout the webcast. To do so, please type your question in where it says type your questions here, and then press submit, and we will answer them at the end of the session today. And now, over to our speaker.
Hello everybody, thank you for listening. Today I'll talk about PCR-free whole genome sequencing using DNBSEQ technology. Let's start with a short reminder. Why is whole genome sequencing WGS so important to sequence for each of our genomes? Genome sequencing is the ultimate genetic test. It's predictive of our future body and mind functioning, and that's why we have to have that genome sequenced. Genome encodes a program for development and adaptive functioning of all our tissues. There is no exception. And because of that, we have to listen, in addition to our body, to our genome, as the cartoon on the left depicts. WGS enables an early pre-symptomatic test, prenatal even, and best interventions. WGS detects all variants, including de novo mutations. For example, using family history, only 50% of cancers can be predicted of female history. But using the genome, we can find those 50% that we cannot predict breast cancer from the family history. So it's really important, and we believe that every child and every person deserves their genome sequenced. But we should keep in mind that predictions are frequently conditional due to environmental impact. There is a epigenetic lock-in, and there are lots of random genetic mutations during our lives. So that's why, in addition to having genome sequenced, we need to do other omics tests to be able to do comprehensive health monitoring. So genome is a complex program. It's an open program, so there is lots of interactions between genome and cell, cell to cell, cell to environment. And all of that is controlling development, adaptive functioning, and aging. So it will take some time and lots of data to understand our genome program. And there are two main types of data. It will be large population genomics data sets, but also lots of multi-omics tests on the differentiated tissues from a large number of edited induced pluripotent cells. So that's a really, really important. We are combining synthetic biology and sequencing here. So once we understand more our genome and how our cells function at the molecular level, we'll have this health monitoring and disease prevention omics tests that hopefully are done frequently. And to enable that, these tests have to be really comprehensive informative and deep with deep sequencing, they have to be extremely accurate. We don't want to predict diseases that we don't have. And above all, really, they have to be affordable to enable this omics for all. And people that are healthy don't pay much money for these screening tests. To enable such types of screening tests, we really need what I name broadband sequencing. Lots of data streaming from our cells to computers. And today, the most efficient technology to do that is massively parallel sequencing, MPS, an advanced form of NGS based on nanoarrays with billions of clonal sequencing templates. So we'll have inherited genome, preferably prenatal test, and then all the other omics and other tests. So what are the critical features that advanced sequencing has to have? First of all, it's always about lower cost, higher throughput, and that enables these populational databases and enables affordable pre-symptomatic screening tests for all. The faster is better in many cases, especially infectious diseases and ICUs. Enables also longer reads in shorter time. Quality is really critical. Higher quality is always better, less errors is always better. To detect rare mutations, no validation necessary if we don't have errors, and no UMIs, no these barcodes are necessary. So longer reads are really sort of important for some applications, full-length amplicons, better genomes, more complete genomes, and then smaller sample is always better, less sample, and easier use. So Complete Genomics is founded in 2005 in Silicon Valley, and we just celebrated 18th birthday with a very focused mission to make human whole genome sequencing routine and affordable using our advanced massively parallel sequencing technology that we call DNBSEQ, DNA nanoball-based technology. From the beginning, with our successes, we catalyzed transition to affordable WGS as the ultimate genetic test. 50 genomes in 2009, then 20,000 genomes, by 2013. Lots of money is invested, over $1 billion during the lifetime of Complete Genomics by now. And now after all of these years of development and many instruments developed, many applications are now based on sequencing, we broaden our missions to driving entire genomics field forward with complete sequencing solutions that we provide to sequencing labs. These solutions improve lives, they help prevent disease and live longer, healthier lives. So DNBSEQ, at the beginning, started with our science paper in 2010, where we demonstrated for the first time that affordable genome is possible, $5,000 genome, when everybody else was about $50,000. And then at the same year, we published first family sequence with the Lee Hood collaboration with Lee Hood. Parents have 2 kids with Miller syndrome. So, demonstrating that when you do whole genome sequencing, you still find a rare mutation, that single base mutations that cause disease. Then we published a paper with that sort of described tumor normal genome to find tumor causing mutations. And then a little bit later, a full demonstration, 50 genomes, patients with intellectual disability, demonstrating the genomes are more informative than exomes. So as I mentioned, since the beginning, the technology developed, we have many instruments, and we demonstrated all, multitude of applications using DNBSEQ. By the end of last year, it was about 4,000 publications. Probably now it's close to 6,000, with a sharp increase in the last 3 or 4 years. And as I mentioned, practically all genomics applications are demonstrated. So let's dive a little bit deeper into sequencing technology. So the Complete Genomics innovations have over 35 years of starting in Serbia. It was 1987 with the first solution for massive parallel sequencing. and more new technologies and innovations are coming. So these innovations are really redefining capabilities of NGS and MPS, and they are covering many areas from the beginning, library preparation, special barcoding, and then array preparation, including our DNA, especially our DNA nanoball array preparation, then different sequencing chemistries. So let's describe a little bit more the most important technology for the massively parallel sequencing, and that's the nanoarray of clonally amplified DNA. So there are multiple approaches. Two of them are based on the PCR, bridge PCR, two primers bound to the surface, and you create PCR clusters. Emulsion PCR using micron-sized beads in oil emulsion. I actually proposed this 30 years ago or so when I was in Serbia as a first massively parallel solution. What we did since then, we replaced PCR with the rolling circle replication, so it's a PCR-free process, and we replaced the beads with beadless process where DNA forms particles, DNA nanoballs that are arrayed on the nanoarrays. So a little bit more details about DNBSEQ technology. Library preparation is typical as in any process. At the end of the library prep, we circularize the library templates, and then we run a rolling circle replication. It's a process based on a polymerase 529 that is very processive, does the displacement, rolls around the circle, and creates this like a concatemer, about 200 copies. We figure out conditions how this concatemer is folded in a small particle, about 200 nanometers. And all of this is done in a single tube, a single valve, no oil, no emulsion. These DNA nanoball particles repel each other. And then we just simply pipette them on a patterned array, nano array, and one nanoball sticks per on a sticky spot that is about matching DNA nanoball size. So what are the advantages of this process? So first of all, this is a, as I mentioned, PCR-free process. We can, circularize 10 bases to 1 kilobase or longer inserts. The particles are really small, 200 nanometers. And as a PCR-free process, because as you can see this, we are always copying the original template. We never make copy of a copy. Because of that, this process has no clonal errors that are unavoidable in PCR process. There is no index hopping also, that is also unavoidable in PCR methods. So because of these things, it's a more accurate way to make arrays and do sequencing. It is also more cost effective. Because the particles are small, allows high density. These particles are bright, allows easier imaging and sort of sharp and bright images. It doesn't overload, so it's a relatively simple, and you can load over 90% of these nanoballs. And at the end, there is no duplication. These nanoballs don't bridge in the neighboring spots, so it's a highly efficient process. So another innovation that we had to implement to use DNA nanoballs, because they are single-stranded DNA, and many times we need to do this parent sequencing to read from both ends. We've developed this, special synchronized controlled MDA process that creates these branches. And interestingly enough, the second strand, to sequence second strand, the second strand is actually has a stronger intensity than the first strand. So, and it can include, regular insert size. So, as I mentioned, the main advantage of DNBSEQ is really high throughput and high efficiency, cost effectiveness. As I pointed out, we already proven that in 2010 with $5,000 genome that sort of started this genomic revolution. And then over the years, we moved and developed more efficient and different instruments for using them in all different genomics applications. I'll point out these two instruments. At the end, we have this highest throughput instrument that we call TX or T20 now. in 2020, we announced $100 genomes. This year, we announced sub-$100 genomes. So it's a really the process that drives to 200 genomes per day and sub-$100 genome. So that's not the end of this technology. We have 500 nanometer pitch on these instruments. We can put DNA nanoballs at 200 nanometers. We can put 4 times more DNA nanoballs, even maybe 16 times. That will drive genome sequence to our $10 genome, will be amazing and enable this routine screens of our health. So talking about complete sequencing solutions, in addition to having instruments that cover low and high throughput, we have instruments that in automation that cover sample and library prep, in addition to sequencing, and then downstream processing data to generate FASTQ files and VCF files. So with 2 goals. One is to have a complete solution for easy implementation, large scale, but also to enable low cost in all steps so that affordable sequencing can be matched by affordable library prep and analytics downstream. So in terms of the quality, as I mentioned, there are no clonal errors, and we can show the one advantage of having no clonal errors, because this is a real PCR-free. When you make PCR-free library, no PCR process in DNB prep, we have a real PCR-free process. We know that PCR causes mostly indel errors, and as you can see here, when we compare with PCR clusters, at the end of the right side, the GATK analysis is about 15,000 errors, and our process is about 3,000. Even with machine learning, we keep about 2 times less indel errors in the WGS sequencing. So it's a really powerful way to do whole genome sequencing. So when we compare the same genome sequence twice, in this case from 2 different libraries, we see there is always a few percent, in this case about 2% difference between 2 runs. But what is nice here, even indels are at a similar level as SNPs. It used to be 10 or 20% for indels. So this process is improved. And why we have those differences? In part because they are somatic mutations. Can we do something about that? Yes, and this is a new process, a new standard in WGS that we call complete WGS. It uses 2 library PCR-free WGS combined with our single tube LFR WGS, implemented on T7 or T20, and provide haplotype phased, more complete and more accurate WGS, and it's affordable, between $200 and $400, all automated library prep and analytics. So how this single tube LFR works. So this is a process invented in 2005 in Complete Genomics, and we published multiple papers in Nature 2012. So what this process does, using massively parallel sequencing, we can barcode all the reads that comes from a long 100 kb fragment with the same barcode, and every other fragment gets different barcode. So in a way, this is like doing long read with NGS. So the first thing is provide haplotyping. So 2 main advantages. This is a process done in a tube, like any other library prep, using the barcoded beads. So DNA wraps around the bead. So that's we have more beads than DNA. So on the right, you see that most of the beads get only 1 fragment of DNA. So this process is low cost because it's a single tube, easy to automate. And because it's a single fragment per bead, it's analogous to single molecule sequencing of 100 kb to 300 kb fragments. So 3 main advantages of complete WGS by combining PCR-free and the single tube LFR. As I mentioned, it's a phased, haplotype phased genome with reduced errors. We detect more structural variants. and also cover some blind zones, some dark genes that standard sequencing cannot sequence, cannot call variants in them. So this is an example of how good haplotyping is achieved by single tube LFR. It's a very good because, as I mentioned, fragments are pretty long, 100 kilobases and longer. So and 50 for the phase blocks is between, greater than 10 megabases, goes all the way to 30 megabases. Some, you can see whole chromosome arms, a single haplotype block, and over almost 99.9% of all heterozygotes are phased. So why is haplotype phasing important? Without haplotype phasing, we really don't know which proteins we have, which proteins our genome encodes. And we don't know which of these proteins are expressed in sufficient quantities. So you can have 2 mutations in the same coding region, or 1 mutation in the regulatory region or a coding region. So we really need to know, do I have an active protein in high quantity? So to achieve that, because these regulatory regions are megabases upstream or downstream of the coding region, we need few megabases haplotype blocks around each gene, and that's really important for the clinic, but also to understand regulation and allele-specific expression, especially in aging, that we really need to understand. So another advantage of phasing, as I mentioned, when we sequence the genome twice, we see these differences. It's not inherited variants, don't change. So we would like to separate somatic mutations from inherited, and the phasing allows that. Somatic mutations and some errors will appear as a third haplotype that we can say this is not inherited, and then we can sort of analyze and keep track of those. So it's a really interesting thing. So finally, with these processes and other improvements, when I sequence my genome twice, my inherited genome should be the same. So I mentioned these blind zones and dark genes. So this is one example, SMN1 gene. As you can see, it's not about DNBSEQ or NovaSeq or any of the NGS technologies. It's about pairing 150 short reads. So this gene has multiple copies. We don't know where to map those. So as you can see, there is no mapping to this gene with the regular sequencing, but with single tube LFR, with these barcoded reads that we know that which 100 kb they belong, we can cover specifically this gene and finally call variants in this gene. And there are many more of these genes, about 20, 30 of these dark genes that single tube LFR process of complete WGS process can now solve. So in addition to whole genome sequencing, having the efficient and cost-effective massively parallel sequence enable other applications. One of them is how to make affordable, deep, cell-free DNA sequencing. It's really important for early cancer detection, for detection of pathogens and the identification of those sort of hidden infections. And we need millions of cell-free DNA tests on millions of people year after year, so we understand that. Hopefully, we can do 100x to 300x coverage, whole genome sequencing from cell-free DNA. Hopefully, that will be less than $100. Once we understand it and we have this conclusion and we generate AI reports, we can do frequent, hopefully annual checks. So for monitoring our health and preventing diseases. 2 examples in this recent publication in Nature Communications, using machine learning and combining the genetic and epigenetic features. This paper shows that at 95% specificity, using whole genome sequencing from cell-free DNA, the DNBSEQ can achieve 91% sensitivity for multiple cancers. So it's a really great example. And with deeper sequencing, it'll be even better. Another application uses panels, capture panels, but it's on a large number of patients, 10,000. On average, 73%, in 73% of patients, circulating tumor DNA is detected. But in a couple of cancer, lung cancer and prostate, 91% sensitivity is achieved. Another really important application of deep sequencing is single-cell sequencing, and especially for monitoring immune systems. If we can sequence PBMCs, the immune cells from blood, 10,000 of them, we be able to monitor our immune system much better than by any other technology. And this is actually demonstrated in Nature Biotech in 2021. Lee Hood's team showed that people infected with COVID, they did multi-omics analysis. They proven that having single-cell RNA-seq enabled them to separate 7 metabolically defined clusters from CD8 plus T7 cells. And that allows prediction of disease severity in patients infected by COVID-19 by monitoring change in cell number in different clusters, subclusters, or change of the gene activity in some of them. So can we do this affordably as an annual checkup? I think sequencing will be affordable, be sub-$100 genome. We need actually to develop scalable single-cell barcoding. And on the right, you see one solution that doesn't require emulsion. It's based on the gel, in this, almost a single tube approach with 10,000 cells in every tube. So when we have these technologies and we integrate them with complete WGS and microbiome, I think we'll be able to really monitor our immune system that is so important. Probably 90% of all diseases are affected one way or the other by immune system. So to do these advanced applications, we need advanced sequencers. And T7 sequencer is one of the ideal sequencers to enable all of this application. WGS, especially complete WGS, cell-free DNA, single-cell RNA-seq. It's fast. It's 24 hours per N, 150 on a large scale, 60 genomes per day. The throughput is flexible, and use of this is flexible because it has multiple flow cell. I'll talk a little bit more about that. And it's really cost effective. This sequencer can sequence a whole genome for $150, and you don't need more than 15 genomes to start the process in one flow cell. So the flexibility is really important because sometimes there are not enough samples and there are different applications. So this is a unique sequencer that has 4 flow cells. Usually sequencers have 1 or 2. It has almost 6 billion reads per flow cell and 23 billion in 4 flow cells. What is really important, each flow cell can be run independently, can be inserted in the instrument independently. If the first one is running, you can still add the other one. It minimizes sample batching. You need only 15 genomes to start the process. You need also similarly 15 cell-free single cell test, because that's approximately the same as sequencing one genome. So you can load flow cell anytime with different samples and even a different parent sequencing structure. So we obviously covered lots of grounds. We covered, there are lots of references that we are citing. It's really sort of a pleasure, and we're excited that we can bring this efficient sequencing technology to enable this advanced application to move genomics forward, so we can understand and monitor our molecular health, and then prevent disease, hopefully enabling longer productive lives. Thank you.
Thank you very much, Rade, for that presentation. It is now time for our question and answer session. To ask a question, please type your question in where it says type your questions here and then press submit. Okay, thank you. We are back in the studio and I will move to our first question. Are the application reagents different from PCR reaction components that you use in your technology?
So if I understand the question, it's about DNA nanoball preparation versus PCR. Obviously, we use different enzyme. It's a rolling circular replication with 529 enzyme. So we have our kits to prepare DNA nanoballs, to prepare circles and DNA nanoballs, and then obviously to load them and sequence them. So from that point of view, they're different. But we can convert any standard library used on any other sequencers just by circularizing and then sequence on our sequencers.
Okay, so that, and then that kind of leads us into the next question, which you have alluded to in your webcast, but perhaps if you can just elaborate on it. What are the advantages of DNBSEQ? So that is the RCR, the rolling circle replication, over traditional bridge PCR.
Yeah, that's sort of many times the main advantages are lost. People think it's just amplification, but it's fundamentally different because in PCR, as everybody knows, it's an exponential amplification. We make copies of a copy, 2, 4, 8, and so on. So whenever there is an error, when we make a copy, that copy with error is propagated. So PCR unavoidably creates clonal errors. In rolling circle replication, we always copy the original template, the original circle. We never make copy of a copy, so there are no clonal errors. There is no mechanism of creating clonal errors. Similarly, there is no mechanism of creating this index hopping, because there is no way to transfer a primer with a barcode from one molecule to another. So these two advantages lead to more accurate arrays without any clonal errors, without any index hopping. So that, fundamentally leads to better data. Even, 100% accurate sequencing, if you have already mutations created by the DNA preparation process, you're sequencing, errors, not the real things from the sample. So that's in terms of the accuracy. In terms of the efficiency, these DNA nanoballs are so small, 0.2 microns, about 200 nanometers in size, but they are bright. In a way, there is enough copies in a small volume, so we can create high-density arrays that use minimal amount of reagents, so it's cost-effective, and also can use cost-effective instruments because we get very sort of bright and sharp images.
I mean, I'm really struck by the rolling nature of it, the fact that you're not making a copy of a copy, and the strength of the technology lying in the copy of the original all the time. Therefore, you don't get the error. Therefore, you get this bright image. Are you able?
Just to add, the sort of everybody knows that PCR creates especially indel errors, because the indels are made usually poly A, poly T stretches, so the polymerase, there is a slippage back and forth. So that creates lots of indel errors in whole genome sequencing. That's why people switched from the PCR libraries to PCR-free. But actually, the process is not PCR-free. If you use PCR clusters to make arrays. Our process is really PCR-free from the beginning to the end, PCR-free library, PCR-free DNB making. So we have about 2 to 3 times less indel errors in WGS.
Fantastic. Thank you very much. I'm going to move on to the next question, which asks, how many whole genome sequences can be run on a T7 per run? And how flexible is it? running a 4FC independently?
Correct. So T7 is a unique sequencer because it has 4 flow cells. Usually other sequencers have 1 to 2. And these 4 flow cells are completely independent. It's almost like having 4 independent sequencers next to each other. So each flow cell, we can run 15 genomes. And the instrument is so fast, these 15 genomes are completely finished in 24 hours. So every 24 hours, if you run only one flow cell, you get 15 genomes. And the reagent cost is the same whether you run one flow per genome, whether you run one flow cell or all 4. So that's really important. So people are not paying higher price because they're running only one flow cell. So they don't need to wait for batching more than 15 genomes to run one flow cell. At the same time, they can run something else anytime later. You start first flow cell, 8 in the morning, and so around noon you have another one. You just plug it in. There is no stopping of the instrument. You can run even a different read structure in the second flow cell. As I mentioned, it's practically like having 4 independent sequencers next to each other. So when you run all 4, it's a very powerful instrument. Sequence 60 genomes per day. In 24 hours, you complete 60 genomes.
Okay, that's great. And it sort of leads us on to the next question, which also relates to the last question about it's about DNA nanoballs and flow cells. And there are alternative companies that are using DNA nanoballs and growing them in the flow cells. What is your view on that, and whether or not that's advantage or disadvantage?
It's a very good question. And actually, I forgot to mention the reason we invented DNA nanoballs was to make pattern arrays with what we call high occupancy. Because in a way, there is no simple way, or almost there is no way to, if I put one circle per spot, to cover all spots to have one circle in every spot. Usually Poisson stochastics says 30% have no circle, 30% have multiple, and only 30% is a single circle. So you get clonal amplification. So any amplification on the surface has this limitation. So you have a less efficient pattern array. With DNA nanoballs, we make these particles in solution. So now, because they're particles, they're not molecule anymore, they have a size about 200 nanometers, we can create sticky spots. So only one DNA nanoball can land on one sticky spot with a similar size. So there is no, rarely multiple DNA nanoballs per spot. So we get 90% or 95% single clonal amplicon per spot. So high efficiency is really important for the low cost.
That's important clarification, I think. So thank you for getting to that question. Our next 2 questions are kind of more application based. The first one asks if we can use this technology for plant genome applications.
Yeah, absolutely. As I think in one of our slides, we have practically any application is demonstrated using DNA nanoballs, DNBSEQ. There is no difference from any other NGS. You make a library and then you sequence it. Maybe the question is related to the single tube LFR because that technology allows to do this like equivalent to longer reads. Some plant genomes are complex and have many chromosomes and many repeats. So there are many papers using single tube LFR to sort of help do de novo assembly of complex plant genomes.
Okay, thank you very much. And our next question asks about poultry diseases and if there is any kits or references that could be useful for poultry disease.
Not sure that we have specific kits from Complete Genomics, but if they're thinking about like panels or something like that, as I mentioned, we can use any panel that is designed for any other platform. We just, have a conversion step, forming circles, and that's not additional step. It's necessary for our process anyhow.
I see. Okay, thank you very much for that. Our next question asks more about product availability. Are all complete genomics products offered in the United States, even the room-sized T20?
Yes, they are, and actually there are even first installations of T20. Yeah, it is a bigger instrument, but it sort of, if we would put together all the smaller sequencers that we need to sequence 50,000 genomes per year, we'll fill the same room, more or less. So it's a really powerful sequencer. For the first time we have a sub-$100 genome. That's why people that have high throughput, and we have a first installation in US.
Okay, thank you.
Yes, all other instruments are available. T7, G99 is a sort of instrument that is, first reports in 3 hours. So, very, very efficient, fast sequencer, probably the fastest in the industry for infectious disease detection. And T7, I think T7 is a sweet spot, with flexibility and $150 genome. It's a great instrument.
Okay, thank you very much. A quick reminder to our live audience, you do still have time to ask a question. To do so, please type your question in where it says type your questions here, and then press submit. Our next question is about costs, and it simply asks, what is the cost of sequencing a genome? I'm not sure if you're able to answer that in full, but perhaps you can shed some light on it.
Yeah, we already sort of talk about cost and efficiency, because it's really important, and the main benefit of low-cost sequencing is expanding applications. Many times people think like, oh, competing on cost is not good, but actually this is not competing on the cost. It's enabling applications that are unaffordable with high cost sequencing. So my personal goal is to get to the unlimited sequencing. So in a way, everything else is more expensive than sequencing. So people can do any depth that they need. So our most efficient cost sequencing is on the T20 sub-$100 genome. So you get 30x genome coverage, standard and high quality. This is not cutting corners on quality. The high DNBSEQ quality for sub-$100. On T7, that is sort of much more flexible and allows you to sequence only 15 genomes in a batch. It's $150. This is really unbelievable. So we also have this complete WGS to do phased genomes. It requires 2 libraries, so it's like 60x genome. But when you have $100 genome for standard, and 2 of them is still, really, almost less expensive, this phased genome, $200 on T20, than many other providers have standard genome. So our goal is really to, with the low-cost sequencing, to move whole genome sequencing and other application to a higher standard. And phased genome. So everybody knows that today, once we announce $100 genomes, many companies, are moving from panels and exome to genome, because it's more informative, it's easier to standardize the process. It was only a matter of cost. The same thing will happen with the phased genome versus not phased genome. Everybody knows that phased genome is better. It's just a matter of cost. And we're solving that with our T7 and T20 low-cost sequences, so we can have affordable phased genomes.
And I'm just going to tease you out a little bit on that by asking you, who do you see benefiting from whole genome sequencing? Maybe first, because I think there's a benefit for everybody, but in the immediate stages and then in the longer term.
It's a very good point. Yeah, you're right. Every genome should be phased genome. It benefits every analysis. Obviously, needs tools to use that information, and they're already developing and so on. So, if you have, there are examples for example, in the IVF. So if you have phase genome of mom and dad, you can better assess which variants the kid has. So, any of the rare disease would immediately benefit because we would know, where that variant that is sort of causing disease, in which context that variant is. Both genes are disrupted, or one is disrupted twice. So, and then also the regulatory variants. So there are many complex situations that phasing will benefit. Or complete WGS will provide benefit. In addition to phasing, the solving these 20-30 blind genes is really beneficial, because otherwise you have to run separate tests to get MSN1 gene or other genes covered. Because when you're doing clinical genome, even missing one gene is not complete. It can be the problem can be in that gene. So yeah, the clinical genomes always should be the highest quality and the most complete. But research is also benefit, as I mentioned, the allele-specific expression is very important to understand. Practically all that we're trying to understand in our genome program, our program of humanity, if you like, is these networks of regulations. How we get differentiation, and some genes are active in one cells and not in the other. And aging is actually a form of differentiation. We just don't want that direction, don't like it, but it's happening. So yeah, we should all switch to phase genomes and complete genomes, and it's sort of the understanding is so complex anyhow, so we need the best data.
Absolutely. And then if I can take it one step further, who specifically benefits from the complete end? That is the double, the double technology that you use of, the complete whole genome sequence.
Yeah, that's phased, and we do PCR-free, our real PCR-free, so it's really the highest accuracy already, and then add the phasing LFR. So, as I mentioned, every the rare disease, any clinical should immediately adopt this, because you don't want to sort of do partial analysis and then go, oh, I didn't find the answer, let's go next time, or a wrong answer, wrong hypothesis. But soon will be standard for all genomes.
Thank you very much for answering that question and being so thought-provoking as to where this direction of travel lies. I'm going to take you back now because we've got some more specific questions on the technology. The next question is asking about FFP blocks. And it's asking about the performance of DNA derived from FFP blocks in this sort of technology. What are your thoughts on that?
Yeah, the standard DNBSEQ, if we don't talk about single tube LFR needs long fragments, so the FFP usually fragments are short, so it's not applicable. But we've proven DNBSEQ can analyze as any other technology efficiently DNA, RNA extracted from FFP samples, because we can circularize any size of DNA from 10 bases to 1,000 bases and sequence highly accurately. So no difference, and it's only beneficial.
Yeah, fantastic. Okay, great. The next question is asking about minimum concentration required for the flow cell. So it's asking, can this be used for targeted regions that are low in copy number in a given sample?
Yeah, so the low copy number in the sample, so people usually solve it by PCR, so they have overall enough DNA, and then sequence as deep as possible. So one advantage of this approach, I mentioned that DNA nanovolts, we can, we have special procedures. We can load almost 90% of them that you supply to the flow cell. So there is almost no much of the loss of DNA. So there is a possibility to do cell-free DNA, for example, PCR-free, especially if it is a whole genome. It's not a panel. Whole genome sequencing of cell-free DNA. Imagine doing that PCR-free, no any UMIs needed, and then the sequencing is so accurate, so you get sort of you believe in every molecule. You can do even per N 200 because the cell-free DNA is short, about 170 bases. So per N 200 can completely cover each molecule on both strands. Practically no errors in sequencing, no errors in the DNA preparation because of PCR-free from the beginning to the end.
Okay. Thank you very much. Our next question asks about how the T7 compares with other systems that are currently out there on the market. There are two mentioned here, but how would your T7 compare with other systems?
Yeah, so T7, it's already 4 or 5 years on the market. Was a leading sequencer in terms of the throughput, 60 genomes per day. It's actually still the fastest high throughput sequencer, 24 hours turnaround time for per N 150. I think the other instruments in this class of high throughput are more like twice longer time. So it's fast, flexible, but overall can produce large data sets to 60 genomes, and it's, to my knowledge, the lowest cost of the genome, $150 per 30x genome.
Fantastic.
So it's a really great instrument for any labs. So the idea, it used to be T7 would be like a high end, but now we want deeper sequencing for cell-free, for single cells. Lots of reads are needed, 10,000 cells. For spatial omics, it requires so many reads that actually now T7 is becoming a sort of like a main instrument in almost any lab.
Okay, thank you very much. The next question is asking about LFR, and it asks, if I understand the question correctly, what you think about using LFR for GC-rich repeats. Can you speak to that?
Yeah, if I understand that question, there are I think a triple repeats, expanded triple repeats, if that is related to that. So the single tube LFR finally reads a per N 150. We have some technologies that will expand the read length to let's say 500 bases or longer. But the expanded repeats are local. So we really need, if we want to measure them, we can measure them with standard sequencing by counting reads, and there are methods for that. But if we want to measure them exactly, we need longer reads. And what is nice about DNBSEQ, we can get into 500 base reads to measure these expanded repeats, but not with single tube LFR itself.
Right. Okay. Thank you very much. The next couple of questions, I'm just bearing with the time here, asks about sequencing. So it's asking, can this technology sequence 10 KB libraries?
We can obviously circularize 10 KB if that is the question, and the longer the circle, the less copies we have in DNA nanoballs. We're making them bigger and bigger to accommodate these longer inserts. So right. in principle, it's possible. We didn't test anything longer than about 1 kilobase.
Okay. And the next question asks what the average sequence file size is after sequencing, and if the process of data analysis is the same as in the traditional pipeline, which would be in a PCR pipeline, I suppose.
It's absolutely the same FASTQ files, the same VCF file, no difference. Even for the complete WGS, we have first automated library prep, so to make 2 libraries, and at the end we have our own pipeline to produce this phased, more accurate, more complete VCF file. So from the user's experience, no difference at all. Finally, it's a VCF file.
Okay, that's great. That's good to know. And the file size?
Same as any other, 30x genome, no difference. Obviously, with more sequencing, bigger files.
Okay, this is very true. All right, on that note, I think that is where we are going to finish for today. So I would like to thank you very much, Dr. Rade Dermanas, for being with us today and for answering all of our audience questions. Thank you very much.
It's my pleasure.
I would also like to thank our webcast sponsor, Complete Genomics, and of course you, the audience, for being with us today. You will be redirected to a short survey, so please do take a moment to complete the feedback questions. You can watch this webcast again also on demand at any time at nature.com/natportwebcasts. Thanks for watching, and I hope you'll join us again soon.